Okay, Naveen, we'll be seeing about the major experiments in hematology.
Yes, ma'am.
So, as you know very well, we have RBC count, WBC count, and DLC as the major experiments,
Yes, ma'am.
each carrying 20 marks. Okay, one among these three will be given to you in the exam. The principle of this cell count is actually we have blood, we take blood, we dilute it with the known volume of the diluting
Mm.
fluid. Okay, and we spread that diluted blood over the Newbar chamber, and we count the cells in a particular squares of the Newbar chamber. From that we'll calculate the number of cells. Okay, and after adjusting for the dilution factor, we will be getting the number of cells that are present in the undiluted
Mm.
blood. This is the
Yes,
same
ma'am.
principle in all the cell counts. Okay, okay, we'll see about the RBC count first,
Yes, ma'am.
right? Okay, how the sample is collected and what sample is collected for RBC count?
Capillary blood using the
Okay,
finger prick method.
from the sterile finger prick.
Yes, ma'am.
Okay, fine. What is the diluting fluid that you will use for RBC count?
Hemes fluid.
Hames fluid.
Hemes fluid. Yes.
Okay, why I am stressing this because many students will mispronounce it as Hemes
Mm.
or Heum or sometimes even Hamas fluid. Okay, so pronounce it rightly.
Yes, ma'am.
Hames fluid.
Hemes fluid.
Okay, can you tell me the composition and function of Hames fluid?
Yes, ma'am. So sodium chloride to maintain the isotonicity, and sodium sulfate it prevents the aggregation of the
Mhm.
cells, and mercury chloride it acts as the preservatives and antifungal and antimicrobial, and finally the distilled water as a solvent, ma'am.
Okay. So in the textbooks or in your practical record notebooks, the grams or the weight and
Yes,
the
yes, ma'am.
quantity will be given. Okay. Many students will try to remember the quantity and the numbers, all the
Mm.
numbers, but that is not important.
Mm.
But what you should remember is the
Function.
purpose of that particular ingredient.
Yes, ma'am.
Okay. So don't forget the ingredient and just remember the quantity.
Yes, ma'am.
Okay, good. Any other diluting fluid that you know of?
Daisy solution.
Very good. If not, none of the diluting fluids are present, what can be used?
Normal saline.
Yes, very good. Because RBCs will be quite viable even in the normal
Normal saline.
saline.
Yes, ma'am.
Right. Okay. What is the dilution factor?
So dilution factor in case of RBC is 1 in 200, ma'am.
What do you mean by that?
The blood is drawn up to 0.5 and the dilution fluid till 101. So the dilution takes place in the bulk portion whose volume is 100.
Okay.
So the dilution part, so 100 part of the diluted blood contains 0.5 part of the blood. So the dilution is 0.5 in 100. So thereby 1 in 200, ma'am.
Very good. Excellent. You have to answer the same way.
Yes.
Okay. In few textbooks, they would have mentioned it, or they would have simplified this as initial volume by final
Yes, ma'am.
volume.
Yes.
All those stories, if you understand that clearly, you answer. Otherwise,
Yes.
you don't answer. Very simple, as you explained in a beautiful way. Bulb portion contains 100
100.
parts. In that 0.5 is blood. So, what happens if you remove the decimal? It becomes 1 in 200.
200. Yes, ma'am.
Okay, that's good. Okay, okay. After dilution, how will you charge the Newbar chamber?
First few drops will be discarded, ma'am.
Okay,
After that, we'll charge into the Newbar chamber.
okay. You put the cover slip
Cover slip.
and you
Yes.
charge. Why you should discard the first few drops?
The first few drops contains only the diluting fluid, ma'am. There
Okay.
won't be any cell in there.
Okay, very good. Okay, fine. Okay, so you have diluted, you have charged. Now the cells are settled under the cover slip.
Yes, ma'am.
Now, how will you identify the RBC square?
So the RBC squares are identified by the triple line borders, and there will be 25 medium-sized square, ma'am. Each
Okay.
medium-sized square is divided into 16 small squares, ma'am.
Okay, so which square you will count?
So for a calculation purpose, we prefer the 4 edges, the 4 box in the edges, and the central one, ma'am.
So 4 corner squares and
And the
the
central.
center square.
Yes, ma'am.
Okay, so does that mean that the RBCs will be settling only these 4 squares?
No, ma'am. It will be spread entirely throughout the
It will
rulings.
be spread over the entire
Rulings.
Newbar rulings.
Yes.
Okay, fine. Okay, So what objective lens you will use for counting the RBCs?
40x, that is the high power, ma'am.
It won't be visible in the low power?
It can be visible, but more eye strain will be there in that one.
Okay, fine. Okay. So now that you have counted in all the 4 squares and the one middle square,
Yes,
okay,
ma'am.
you'll have to note the numbers
Numbers.
specifically. And one tip I will tell you, the numbers in each medium size square after calculating the total, it should not differ from the other box. It should not be more than 10.
Okay.
Say for example, right top square, left top square. If you take these two squares, here if it is 90, here it should be either 80 or 100. So the difference should not exceed
10.
more than 10.
Yes.
If it is more than 10, so this should be congruent with all the 5 squares. If any of the squares are having the difference of more than 10, then that means the cells are not spread uniformly.
Oh,
It's an indication, rough indication.
okay.
So make sure that the cells are within the difference of
10.
cell
Yes, ma'am.
counts from each box. Okay,
Yes, ma'am.
fine. And you must also know the steps of calculation.
Yes, ma'am.
After that, you will count the total number of cells. Like for example, N1 is the top left, N2 is the top right. Then N1 plus N2
N2.
plus N3 plus N4 plus N5.
Total
That gives the
N.
total number of
Yes.
cells. And after calculating for the size of the squares and the volume of the squares, all those things, finally you will include the dilution factor of 200 also. Finally the formula will come into n into 10,000. But you have to write the steps of the
Yes, ma'am.
calculation, because examiners will be very particular about the steps of calculation.
Yes, ma'am.
Okay, you should not just directly write n into 10,000. Okay, so that the calculation part is over.
Yes, ma'am.
Fine. Now before that, I forgot to ask you one question about the RBC pipette.
Okay.
How will you identify the RBC pipette?
The presence of markings 0.5, 1 and 101, ma'am.
Okay.
Also will be having the red bulb, red bead, and the bore will be narrow bore,
Okay.
and will having the red mouthpiece, ma'am. So these are the identification points for the.
Okay. What is the purpose of this red bead?
The red bead, it acts as an identification,
Okay.
and it's used to mix the diluting fluid with the blood,
Okay.
and to ensure the pipette is dry.
Okay. Are there any other uses of this RBC pipette? Any other cell can be counted with this
Yes, ma'am. WBC can be
WBC?
in case of leukemias.
Yes. Okay.
Also platelet count,
Yes.
sperm count.
Yeah. Very good. Excellent. Okay. Now what is the normal RBC count?
For the adult male, it is 4.5 to 6 million cells per cubic millimeter. For the female, it is 4 to 5.5 cells per cubic millimeter, ma'am.
Okay. In children?
It will be higher, ma'am.
Why?
No idea. More erythropoietic action, ma'am.
More erythropoiesis, the marrow is fresh, so it produces more RBCs, but at the same time, the RBCs will be lysed quickly also.
Yes,
Their
ma'am.
lifespan will not be 120 days. Okay, it's very short because the liver and the spleen are very immature.
Yes,
Okay, so the count will be more.
Yes, ma'am.
Fine. Okay. Now you said for males it is higher. Okay, why so?
So testosterone plays a major role in the erythropoiesis production, ma'am. Erythropoietin production, thereby increase in the level of RBCs.
Okay, fine. What is increase in RBCs called?
Polycythemia.
Okay. Can you tell me some causes for polycythemia?
In the physiological cause, it is high altitude.
Okay, good.
For the pathological, it is congestive heart failure, chronic renal failure, congestive pulmonary diseases.
Okay, so all these conditions basically they end up in hypoxia.
Hypoxia.
Okay,
Yes, ma'am.
so once hypoxia is there, it is automatically the stimulant for erythropoietin, and that's how RBCs are produced more.
Yes, ma'am.
What is polycythemia vera?
Polycythemia vera is the primary disorder in the hematopoietic tissue.
Okay.
So thereby it result in increase in the RBCs, ma'am.
Sometimes even WBCs and platelets
Yes,
also be increased in polycythemia vera. Okay, good. Fine. What is decrease in RBC count called?
Anemia.
Okay. Can you tell me some physiological causes and pathological causes for anemia?
In children, pregnancy, and
Why in pregnancy anemia occurs?
So pregnancy is the state of hemodilution, ma'am.
Okay. So, in fact, actually, the marrow will be active.
Mm.
Cell count will be more also, but more than the cell part, the plasma part will be
Okay.
expanding more. So, there occurs hemodilution. That's
Mm.
why it appears that uh, RBCs are very less in
Yes, ma'am.
pregnancy. Right. Okay. Any other conditions? Pathological conditions?
Pathological, any type of anemia.
All types of anemia will cause decrease in RBC.
Yes.
Okay, so the most important in RBC count is the diluting fluid, dilution factor, okay, and the squares that you are choosing,
Yes, ma'am.
okay, normal count,
Mm.
increase in the cell count, decrease in the cell count, the causes for both
Yes, ma'am.
these are the key questions. If you answer all these key questions, they may go for other questions.
Yes.
Okay, good.